Hi, and welcome to the third SCiLS Lab tutorial, Data Segmentation. First, I want to explain why we need segmentation and what exactly it is. So as you probably know, manual evaluation of large imaging data sets is totally unfeasible, and it really requires data mining strategies. Automatic spatial segmentation is one of these strategies and can be used as a quick first step in the data mining process. Essentially what happens is similar spectra are grouped into colored expandable clusters called segments. Today I'll start by covering the segmentation pipeline. The segmentation pipeline is only used with axial top data. If you do have other data types, you can use the segmentation tool instead of the pipeline, and this tool can be found under the Tools menu. The pipeline is necessary for axial top data because, as we covered in tutorial 1, axial top data does not contain a peak list. All other data types come preloaded with peak lists. So as part of the pipeline, a peak list is created, as you'll see. We'll also cover how to handle interactive segmentation maps and how to store segmentations for further analysis. First, I want to explain why we need segmentation and what it is. Manual evaluation of large imaging data sets is unfeasible and requires data mining strategies. Automatic spatial segmentation is one of these strategies and can be used as a quick first step. Essentially, similar spectra are grouped into colored expandable clusters called segmentations. Today I'll cover the segmentation pipeline, how to handle interactive segmentation maps, and how to store segmentations for further analysis. With that, I'll open the SCiLS Lab software, where I already have a file open. As I mentioned, I'll cover the segmentation pipeline today. Pipelines are basically shortcuts for important combinations of tools and can be found in the pipeline menu. Select segmentation and you'll see a menu where you need to define a set of parameters for your segmentation pipeline. First, specify how the spectra should be normalized before the segmentation. Only normalized datasets offer reliable results for spatial denoising and segmentation, so this is important. I'll choose the total ion count normalization. If you have MRMS or timsTOF flex data, use the root mean square option. Next, choose the regions that you want to segment. I'm going to choose all of the regions. Now, you need to specify whether spatial denoising is applied before segmentation. This often leads to less noisy segmentation maps. You can experiment with this, but I'm going to start with strong. Now choose a name for your segmentation map and select OK. The data processing will begin immediately in the background. If you navigate to the Tasks tab, you can see the progress. Once it's done, you will get a notification, and you can navigate back to visualization. Now the segmentation map will be stored initially under Labels. So expand Labels, click the drop-down menu, and you should see your segmentation map here. By default, you start with 2 clusters, shown here in red and green. As I mentioned, these are interactive segmentation maps, so you can expand and collapse these clusters as you see fit. By default, double-clicking a cluster will expand it. Double-clicking the parent spectra will collapse it. You also have the option to perform an action when clicking on a label. By default, it does nothing. You can change this to expand and merge with a single click. You can highlight specific clusters in white. And you can completely hide clusters. Related to this, you can make the entire segmentation map transparent. This can be useful when trying to match anatomical features with features in your segmentation map. You can also manually merge different clusters. Say for instance, I would like to merge the green with the gold. Hold Control, select both clusters, and click the Merge Selected Classes button. You've now merged those two clusters into a single cluster. Take note, there is this button to reset this tree to the default segmentation map. Lastly, I'd like to show you how to create a region for further analysis from a segmentation. For example, I would like to choose this yellow region, this yellow cluster to be saved as a region. Select the yellow cluster. Click Save Selected Class as a Region. Name the region and save it. Now, we'll turn our segmentation map off and navigate to regions. You can already see our new cluster is already visible. Our new region is already visible. And we can turn it off and on the same way we turn off and on any region. I also wanted to mention you do have the ability to combine two or more regions to form one single region. You would want to do this, for example, if the regions share histologic or biologic significance. I'll show you an example here where I combine our three pyramid regions into one new region. If you recall, you can click on the eyeball icon next to a region to visualize it. Now, if you hold Control while selecting regions, you can select multiple objects. So in my case, I select Pyramid 1, 2, and 3. Navigate down to this button, Create New Region from all selected regions. You have two options here. You can create a new region from the selected regions, or you can move the selected regions into a new region where these existing regions, Pyramid 1, 2, and 3, will be subregions. So in my case, I'm going to choose the first option. Select a name for your new region and save it. So now, if we turn off our Pyramids 1, 2, and 3, we can see that our new region pyramid has all three of the regions I combined. If you want to delete a region, navigate to the Objects tab. Select the region you want to delete. This pyramid region is the region I just created. And press the delete button.
Thank you for watching. Please open video tutorial 4 co-localized with M/Z values to continue.